Okay, well, good afternoon, guys. I'm sorry I've not been able to make it to class today. Normal service will be resumed next week, but I want to say, first of all, welcome back. I hope you had a really good summer. I hope you're able to take some time off to relax and recharge, and you're ready for the semester coming up now. I've met everyone. We all met during Biochem 1. So you kind of understand a little bit about how I like to run my class, how I like to do things. But today I would like to just go through the syllabus, a few of the kind of expectations of the class and where we're going to go with it and things like that. And then I am going to go into a lecture. The lecture is going to be on enzyme kinetics, and actually it's mostly like 90% of it is stuff that we covered before. I'm going to go over it a little fast, but like I said, we covered it before. And really it's just to set things up so we can move into new material in the next lecture nicely, ready for the semester ahead. The main thing is that I don't want to rush things as things go along. I want to pace things nicely, and the sooner we start, the easier it is to spread things out. So you guys hopefully can see my screen right now, and this is the Blackboard screen. This is my screen, but you should have something similar. If you can see along here, there's going to be some Achieve homework here in some weeks. The lecture slides are under course content. They've all been uploaded. I'm going to upload some questions, which are going to be practice questions, ready for the exam and things. But also I'm going to upload some what I call class questions. These are going to be things that I'm going to talk about for each individual lecture. And it'll be things that you can use to follow along to really help you remember what was covered in this class, what kind of things am I talking about. They're big hints about the kind of content that I'm going to cover in the exams as well. There are of course going to be plenty of quizzes and homework. This is all stuff you won't be able to see any of this yet, but you see that I've already set up all the quizzes and it's going to pop up. So the first quiz is going to be on enzymes. It's going to be on August the 25th, which I believe is Monday. So that's going to be the first one. It's going to pop up at 6 a.m. It's going to go dead at midnight. You need to complete it before midnight. So it's the same as it was last time in Biochem 1. And there's some homeworks as well. There's an enzyme kinetics worksheet, which is, I guess that's going to pop up on the Wednesday, which you need to complete and then upload. There's an essay homework coming up in October that you'll need to complete and upload. Which we'll talk about that when it actually comes up. But all these things are gonna come up as the class goes on. And like I said, there's also homework in the Achieve platform. So please do click the Achieve link and register for the course. Ideally, so that we can know if there's any problems, so we can solve those. You should all have codes for the upcoming semester and access to the textbook as well to help you. But let's just take a look at the syllabus. So I've noticed that I put two things in here. I've got the syllabus and an assignment timetable. And the assignment timetable is literally a week by week, by the 16 weeks, and also the week beginning Monday, whatever, and it'll say whether or not you have a quiz or a homework for that week. So this week there's nothing. Next week, like I said, on the Monday there's an enzymes quiz, and on the Wednesday there's the enzymes kinetic worksheet. It's actually just like the one that you did in Biochem 1, and some of the questions are repeated with new numbers, but kind of they're repeated to try and refamiliarize yourselves with what I'm covering and make you comfortable with the content. But also some new questions as well, some tough ones for some of the content that we're covering. And then the following week, the quiz on bioenergetics on the Wednesday, on glycolysis on the Friday, but then no homework. So it's going to be similar to how it was before. But here is the syllabus for you guys. So we are Biochem 2. We meet at 9:30 in room 207. Here's me in my office. I'm on the top floor. I'm next door. I'm in between Dr. Cooper and Dr. Alex over on the top floor. And please, drop by whenever. The best way to contact me is to email me, and I'll get back to you within 2 days, Monday to Friday. You all know Dr. Sefer is our department chair, and I do have flexibility to meet with you. I know that sometimes people see student hours and think those are the only hours. They're definitely not the only hours. I will make time to see you, but my student hours are the times when I will commit to seeing you. They're the times where I'm pretty much going to be sat in my office just waiting for someone to turn up. I'll probably just be doing some paperwork. If I'm not, just come and knock on the lab door. That's the only other place where I would be. But I try to make sure that I'm in my office unless one of my research students pulls me over and needs something quickly. My lab is 3:14, but I should be in my office 3:39. On Monday, 11 till 1. Tuesday, I lecture all day. Wednesday, afternoon, 2 till 4. Thursday, I lecture all day. And then Friday, I'm kind of in and out of the office. So I like to try and put aside the mornings for student hours, but I don't necessarily commit to it. But so I won't commit to being sat there. But if you email me, I'll make sure I'm there. And any times outside of these times it doesn't work with you, just email me. We'll figure it out. So these are not the prereqs actually. The prereq is 451. But and if we look at the course description, so like we know from before, biochemistry, the chemistry, how does the biology work? The chemistry of the biology, the chemistry of the biological functions. And we already covered Biochem 1. And we covered some of the basic molecules of amino acids, making proteins, lipids, carbohydrates, and nucleotides, those chemical structures, and some of the biology that they took place, biological reactions they took place in. And a kind of a little bit of the reason why the chemistry explains the biology, the biological response that we see. So where we're going to go in Biochemistry II is now we're going to do more application. We're going to largely do a lot of bioenergetics, so the chemical reactions of how energy is made in the cells, because it's just a really handy way of describing, again, the chemistry that makes the biology happen. And really, biochemistry is kind of biological molecules and metabolism. We did a lot of the biological molecules in Biochem 1, so now we're going to do the metabolism. So we split it up like before, exam 1, exam 2, exam 3. It's split up a little bit awkwardly, to be honest. Part of that is just because to try and make the exams balanced. And there's always a question of, well, do I do 2 exams, 3 exams, 4 exams? I'm still kind of playing around with it a little bit with Biochem 2 to find the best way. At the moment, I'm settling on 3 exams, but we'll see how that goes. We have the topics. Topic 1, 2, 3, 4, 5. I've numbered it a little wrong, my bad. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, actually. Exam 1, exam 2, and exam 3. So we're going to start out, we're going to talk about enzyme biochemistry, like I said, and I'm going to talk about that in this lecture. And we're going to revisit the old biochemistry we did in Biochem 1, but also I'm going to kind of, this lecture is actually largely going to be a repetition of a lot of the biochemistry lecture. I'm kind of compressing it all in that kind of 3 lectures into 1 lecture very quickly because we did it before, but to refamiliarize yourselves with it because we're going to build upon it. So we're going to talk about enzyme biochemistry. We're not going to talk about carbohydrates, but I am going to upload the lecture slides again because this is largely for people who didn't do Biochem 1 with me and they may not have approached carbohydrates in the same way. So I'm going to upload the lecture slides from Biochem 1 and they're just there for you to read to help refresh your memory. I'm not going to lecture on it. I'm not going to directly test on it. I'm also going to talk a little bit about bioenergetics. You notice that it's not in this little blob of exam 1. That's because carbohydrates, like I said, I'm not going to exam on it. Bioenergetics, I'm not going to exam on it directly, but the principles of bioenergetics are going to be applied to glycolysis. There is a bioenergetics quiz. There is not a carbohydrates quiz, and neither will be directly tested on the exam. However, glycolysis is the steps and reactions that animals, or actually most life on this planet, extracts energy from sugars, from carbohydrates. And so understanding carbohydrates and bioenergetics is really putting those two together and looking at those reactions, those 10 steps that are very conserved among all life, barely changes between a yeast to a plant to a mouse or a bat to a human. Pretty much the same all the way through. Even the enzymes are very, very similar. They don't really change. They are the same enzyme, but maybe there may be some slight changes to the structure and sequence, but very, very minor actually. And so, you know, indirectly, carbohydrates and bioenergetics are going to be tested because we're going to talk about the structure of sugar, of glucose. How does the structure of glucose change? How do all the bonds move around and things like that? So we're going to talk about those 10 steps. Going to move into anaerobic respiration. So that's like when you're running and you're using up oxidizing glucose faster than you can provide oxygen for your mitochondrial respiration. And so you do that anaerobic respiration where lactic acid builds up and your muscles ache. We're going to talk about that, but also from the point of fermentation, like how beer is brewed and through anaerobic respiration of yeast producing ethanol instead of lactic acid. And a little bit about the industrial practices of that, and things even going into fermentation for like fermentation of yogurts, or for the industrial preparation of propanol, things like that. Then after glycolysis in sugar metabolism, the end product of glycolysis is pyruvate. Pyruvate is converted into either oxaloacetate or mainly acetyl-CoA, which feeds into the TCA cycle. And we're going to talk about the steps of the TCA cycle. And so enzyme biochemistry, so some questions on enzyme function, multi-substrate enzymes, things like that. Glycolysis and TCA cycle, that makes up exam 1. After that, we're going to move across into the exam 2 material. The exam 2 material starts with how things branch off from the TCA cycle. So the point of the TCA cycle is to produce NADH and FADH2, which is really just a carrier for the electrons that have been pulled off of the sugar and are going to be donated to the mitochondrial oxidative phosphorylation reactions for the production of ATP, which catalyzes a lot of chemical reactions in the body. And so the TCA cycle, it can actually go in 2 directions. It can go forwards into oxidative phosphorylation in the mitochondria producing ATP, or it can actually go backwards, do reverse glycolysis called gluconeogenesis, which is sometimes done when we need to go backwards because we need to look after some, we need to reproduce glucose because we've kind of maybe used too much, or like the liver needs to create a store of glucose that it can supply to the body. So we're going to talk about that in more detail. And then we're going to talk about photosynthesis, because photosynthesis is actually very similar to oxidative phosphorylation. And that's kind of interesting. And we're going to talk about the evolutionary link between them there. So that's going to make up exam 2. Exam 2 has got an essay. So exam 1, it's all MCQs. Exam 2 has got some MCQs, but also a few, 3, I believe, essays in it. So we're going to talk about that, and actually the homework is going to be to do mock essays. So that's going to really prepare you. Exam 3, we're going to move into fatty acid catabolism. So that's how fatty acids are broken down to supply energy, and a lot of that is going to feed into oxidative phosphorylation actually. And also amino acid metabolism, so that's how amino acids are broken down, and the amine of amino acids goes into urea. So we'll talk about the urea cycle, goes into urea, which is then excreted in urine, and then the rest of the amino acid, once you pull off the amine group, you just got a carbon skeleton. And that carbon skeleton has got energy, just like glucose, and that can feed into oxidative phosphorylation and the TCA cycle. So fatty acid catabolism and amino acid metabolism, you're breaking down fatty acids and amino acids to feed into either the TCA cycle or directly into OXPHOS, again to produce ATP from a series of redox reactions involving NADH, NADPH, and FADH2 and things like that. So that's how they link together. Exam 2 is also a mixture of MCQs and essays. And we'll cover that as we go along. So I have the student learning objectives and outcomes. These are really saying that I want you to be able to predict what is going on and understand the chemical reactions, and then use that understanding of the chemical reaction to predict how it's going to influence the biology. That's a kind of higher level of thinking. and to also, there's a certain amount of memorization, sure, but there's a certain amount of, well, I know this is true, so I predict that this is going to happen. And that kind of critical thinking is really important, and it's going to be important for your careers and graduate school going on. And so I'm trying to instill those habits. You're going to require the textbook Leringer, same as last time, and also you're going to use the Achieve platform. If you want another textbook, I recommend Biochemistry by Berg or Molecular Biology of the Cell by Alberts. They're both really, really good. They probably have them in the library. And if you want an alternative way of going over things, there is AK Lectures on YouTube. Is really, really good. The guy tends to just cover like individual concepts rather than kind of rigorously going through the material from start to finish. He just picks something like the Corey cycle, the Cahill cycle, you get the idea, the Q cycle. Just grabs them and just talks about them for 20 minutes. He does it a really, really good job. So that's a good way of looking at things. There's also a lady called, I believe it's Elia Hefner. She actually just goes through the textbook that I teach from and goes through the lecture slides and goes through them pretty well and pretty thoroughly. She doesn't expand on points so much, but she really rigorously and thoroughly covers the key points. So she's a really, really good person to watch and listen to pick up things. And also Khan Academy does a certain amount as well. Grading policy, same as before. Grading allocation for 1 hour exams. So exam 1, exam 2, exam 3, and then the final, which is going to be a little bit of everything. Quizzes make up 30% and homework make up 30% for a total of 100%. Exam 1 is MCQs and drawing, just like what you're going to be used to. Exam 2 and 3 are MCQs, drawing, and long answer questions. I'm gonna give you guys practice and work, and I'm gonna help you through the essays. I'm gonna give you the essay questions in advance. However, that means that because I'm giving them to you in advance, I do expect good answers. I do expect good and thorough answers because I'm going to give you what the questions are. Or actually, I'm lying a little bit there. I'm going to give you about 9 questions, 3 from one of 3 different topics, and I'm just going to say I'm going to pick 1 from each different topic to appear on the exam, but you're not going to know which one it is until the exam comes. So you're going to have the questions, you're not going to know which one I'm going to pick. So I do expect fairly good and complete answers, and I will be a little bit tough on the grading there. But the first time it happens, there's going to be a homework, you're going to get feedback, we're going to work together, and I want you to have good essay answers by the time the crunch time rolls around. Like I said, there's quizzes are available on Blackboard. They pop up in the morning, they close at night. Please do complete them. I've been a little bit relaxed with quizzes in the past, where I've let people do them late, but I'm trying to be stricter now, and they will just close and won't let you go in, and unless you have a good reason. I'm not going to reopen them. And you know me, if you have a good reason, you have a good reason. If you were sick or something, I'm fairly relaxed with that. I'm more interested that you do the work than you feel held back and you feel like an illness got in the way or something like that. I just want you guys to do well, but you just got to do the work on time. I'm going to be firmer with deadlines. Similarly with homework, if it's late, I'll just take points off. So please try to complete it on time. Course policies are, like I said, missed work. If it's no late quizzes, homework. If it's late, I'll just dock points. No makeup exams unless required underneath university policy. So that means if you were ill or something like that. Bonus points again, as I said, and that is the main points really. There's the class schedule. So I have what I'm going to cover each day by the different weeks, and when the quizzes are going to be, and the reading material that you can read in the chapters in the textbook. So there's a total of 14 quizzes and 7 homeworks. And I think that covers everything. Please do not use AI for writing things. That is because I want to look at how much you know. You're also kind of cheating yourself because the homework is only worth 30%, and there are 7 of them. Yes, so the essay homework is not actually worth a huge percentage of your grade. The essay homework is only worth like 2 or 3% of your grade or something like that. So if you cheat on it to get 100% there, well, yeah, you get 2 or 3% there. However, then when it comes to doing the exam, it's worth 50% of the exam, and then you're going to get a low grade on the exam because you didn't do it yourself. You got AI to do it. You don't really understand the question. You can't answer it. You bomb the exam, and then it's going to end up hurting your grade so much more. So don't use AI. If you write a mediocre homework essay, then you may only get 2% instead of 3% on your grade. But then what will happen is that you'll get feedback from me, so that when it comes to doing the exam, you'll do so much better on the exam. So it's not worth it. You're cheating yourself. You are, I know people, professors and teachers say that if you cheat, you're only cheating yourself, but really you are, because you won't get that feedback and you'll struggle on the exam. So please don't do that. Now, certainly, I don't want to be a Luddite and say don't use ChatGPT at all. What I'm saying is don't use it to write because you're cheating yourself. But also, I will consider it as plagiarism, and it's really easy to detect, to be honest. ChatGPT and things, they do not write good essays. It's really surprisingly obvious. Also, by all means, like I say, I'm not going to be a Luddite. By all means, use it to help you research, but I will say again, it lies. I've used it to help me research in the past, and I said, hey, I know that X is true. Does that mean that it applies to Y? And it'll go, oh, yes, it does. These are the reasons why. And I'll go, oh, that's interesting. I didn't think that was true. I look into it deeper, and actually, no, it's not true. It's called a hallucination. They call it the AI hallucinates, and it will do that a lot. By all means, use it to help spark creativity and to help guide you along the path, but validate everything because it will lie. And if you say something isn't true, I'm going to take off points, and I'll do that in the essay, in the homework, and in the exam. So and I think that's really it. If there's anything that you would like to know more about, then please let me know. Please, I'm really open to questions. I'm really open to conversation. So you know how things are. I'm really happy to work with you. So please drop by if you want any clarification on anything. But I believe that is everything. Yeah. Okay, so I'm going to go into the lecture material now. I'm going to talk for maybe about an hour with the lecture material, try to cover about 30 slides. I am going to go fast, but that is because this is all material that you guys have seen before. And also it's a video, so you can just play in halftime if you want to. But we're going to go through enzymes, and these slides should all be familiar. I believe that these 2 slides are new, and we're going to go through up until here, which again is stuff that we covered, and then we're going to go into enzymes that catalyze 2 or more substrates. So you're used to a substrate binds with enzyme, produces products, but there's only one substrate producing one product. That's only about 1/3 of enzymes actually do that. About 2/3 of enzymes actually have one thing binds, a second thing binds, one product leaves, a second product leaves, something like that. And there's a whole different order of it actually. So we're going to cover that in this lecture. We're going to cover that on Tuesday. We're only going to get up to slide 35. Like I said, I'm going to ram through it pretty fast, but this is all material that you should be familiar with, and it's a video, so you can play it a little slower if you need. So, enzymes. So you're familiar with enzymes from before. They are proteins. Usually, there are actually some enzymes that are made out of RNA, but that's getting a little bit out there. But they are proteins. They are fully folded proteins in their native state that act as catalysts. They produce a way of making a chemical reaction go forwards by adjusting. Well, they lower the activation energy by adjusting the equilibrium constant. But we're gonna talk about that as we progress. So with this lecture series, I'd like you to understand what an enzyme does in terms of the energetics, the thermodynamics, how enzymes bind to their substrate, because you need to bind with the substrate in order to make the chemical reaction happen. Understand these constants, the V0, Vmax, Km, and Michaelis-Menten equation that we, like I said, covered in enzymes in Biochem 1. But I'm going to refresh your memory here. And then these reciprocal Lineweaver-Burke plots. You remember that the enzymatic reaction has got a curve, but sometimes we like to instead use a straight line. I remember there was probably a question on it on the exam with the straight line. And it may not have really seemed super important in Biochem 1, but Lineweaver-Burke plots become a lot more important in Biochem 2, because with inhibitors you can overlay them and you can work out what type of inhibitor it is from that. And that's what we're going to cover on Tuesday. And then on Tuesday we're going to cover the competitive inhibition, and actually says know that there are other ways of inhibition. Actually, we're going to cover those other ways. We're going to cover the uncompetitive, the non-competitive, the mixed type inhibitors, which are the other types in addition to competitive. And then same with the permanent inhibitors. We're going to cover those. And then we're going to go through a few case studies as well, which will probably be on Thursday next week. So why are enzymes important? So like I covered last time in Biochem 1, they are a way of selectively making a chemical reaction occur. Because if you have a substrate, this substrate may chemically react to produce a few different products. But by using enzymes, you can selectively just make the products that you want, because the shape of the enzyme will guide it towards the product that you want. So you get selectivity. You get selectivity of the substrate that is used, and you get selectivity of the products that is produced. Let's say that you have a chemical reaction that's catalyzed by just sprinkling in some magnesium. Well, that magnesium is going to catalyze all reactions that it can catalyze, and you don't know what product. It may be a mixture of products, and also as well, it's going to, as long as the magnesium is there, it's going to be working. With enzymes, often the enzyme is actually just a protein that's enveloping that magnesium, and so it can control what goes in, so you can control the substrate. The protein has a shape, so you can guide the reaction towards one particular product, so you can control what goes out, but also you can open and close the enzyme, for instance, so you can turn it on or off. So biocatalysts actually have an advantage over regular inorganic catalysts. I gave the example of a metal ion envelops in a protein, but there's all sorts of different ways that this can occur, depending on the catalyst. So like I said, many things may bind for the reactant to make the product, or rather the substrate, same thing, but we use the term substrate. Substrate can make the product, but maybe if it's the wrong substrate, so I believe that this, yes, so this is the enantiomer. So maybe the L will bind, but the R will not, the D will not. So it's you can control which one reacts, and this one will not even bind, or maybe this one will bind, but nothing will happen. So the reaction can be summarized as the enzyme and the substrate come together and bind to form an enzyme-substrate complex. They then react. There's a chemical reaction that occurs to turn it into an enzyme product complex, so the two are stuck together. And then that complex is going to break apart as the enzyme releases its product, but you start with enzyme and you end with enzyme. So you go back around again. Now, in between, you have the enzyme bound to something, so you have these intermediates, but you start and end with the same thing. Sounds like a catalyst, right? And we're going to talk about really the kinetics that control this, because notice we've got these double-sided arrows here. That's saying, remember, that's an equilibrium. And remember that in equilibrium, if we've got substrate to product in equilibrium, that we can actually, this equilibrium has got a symbol K, and we can quantify K as being products over reactants, or let's call it substrate, because we are talking about enzymes, so we use the term substrate, same thing but just terminology. And so this K tells you, well, does the reaction prefer the product or the substrate? And so if you have a chemical reaction of, let's say, of S to P, well, it may be that without enzymes, the K is low. What enzymes do is that they make K high. Because if K is high, if this number is high, it's saying that you have lots of this. And so it's saying that the reaction is proceeding forwards. If K is low, such as in the uncatalyzed reaction, then, where's my pointer? There it is. If we're saying that K is low, then what you're saying is that substrate is, this number is high and this number is low. So you have lots of substrate, not much product. Well, that means that the reaction is in this direction, and it's not really proceeding forwards at all. So enzymes increase K to push the reaction forwards. They're making it so there's more product than the substrate, and I mean, if you start with no product and you're ending up with some product because the equilibrium shifted, that's the reaction going forwards. So that's what, and we're going to talk about how we measure all of these. Like I said, I covered that in Biochem 1. I'm going to recover it again now, so you guys are ready for Tuesday's lecture when we're going to go over things, and ready for when we start talking about this applied in glycolysis. So, like I said, the general thing is that if we've got a reaction of going from S to P, we know that there's going to be a transition state where there's kind of, and this transition state is given by the symbol, this double cross here. And it may be, for instance, let's say we've got 2 carbons and we've got an OH here. Let's do it like this to make it a little bit cleaner. We've got an OH here, and this is the substrate, and the product is where we have the OH here. So it's moved from 1, 2, 3. It's moved from carbon 2 to carbon 1, for instance. I haven't drawn in all the hydrogens. You get the idea. And so that may be the chemical reaction. And it may be that the transition state is, for instance, run in our room a little bit, some kind of diol, for example, something like that. And actually we're going to talk about where that is the case. Actually, that is the case of the intermediate, some of the chemical reactions we're going to talk about. And so what happens is that you get the OH on one carbon, and then there's the intermediate of the OH being on both carbons, and then the OH gets taken off one of them in order to give you the product you see here. So as the substrate to product, you have a transition state, but the thing is that this molecule maybe doesn't want to have 2 OHs on there, and so actually it requires a lot of energy to get there. What enzymes do is that they create an alternative transition state that is probably, in most cases, more stable. Because it's more stable and it's easier to get to and achieve, less energy is required to get to it. And what you're actually doing there is that you are moving the equilibrium. When you do that, by providing that alternative path, you are changing the equilibrium constant, whereas with the original path, K is going to be low, but with this new path, K is going to be high. And what we're describing is that the energy required to meet the transition state, well, that's going to be the energy required to get to the transition state is going to be delta G transition state, and uncatalyzed is going to be uncat, and then catalyzed is going to be cat, and you'll notice that uncatalyzed, the number is bigger than catalyzed, so less energy is required. Delta G cat is smaller than delta G uncat, reflecting that less energy is required in order to hit that transition state. And often it is the case there is several small bumps instead of one big bump. This is oversimplified, but there may be more transition states, but they are easier to achieve in a enzymatic reaction. And just to be clear, sorry, on the left axis there's the energy state, and on the x-axis that is the reaction coordinate, which just effectively means time. It's the reaction proceeding. So like I said, enzymes are proteins. They need to be fully folded, and structure equals function. If something happens to the protein to change its shape, it's going to change its function, and that change may be on, off, fast, slow, substrate 1, substrate 2, etc., etc. And that's going to come up more when we talk about the applications. I believe that we covered before that often an enzyme, I keep losing my pointer, where it is, there it is. Often an enzyme, shown here, requires another molecule to bind to it, a non-protein molecule to bind to it to help it do its job. And these are referred to as cofactors. The other molecule is a cofactor. And cofactors can be split into two groups: inorganic ions, such as iron, magnesium, manganese. So the enzyme on its own, termed the apoenzyme, is inactive. It requires the metal ion to bind to it, an inorganic ion cofactor to bind to it to help it do its job. And when it is bound to its cofactor, it is referred to as the holoenzyme. So apoenzyme is empty, holoenzyme is complete. And so, like I said, for instance, there may be a chemical reaction, a standard chemical reaction that is catalyzed by magnesium that you could do in the chemical synthesis labs upstairs. But if you get that magnesium and envelop it in a protein, then that protein can control switching it on and off. And when that protein is bound to the magnesium, it is the holoenzyme, it is complete, able to do its role. The other type of cofactor, which is an umbrella term, I'll just write cofactor, cofact. You got the inorganic ions in org, and you have coenzymes. Coenzymes. So it is, a coenzyme is a type of cofactor. An inorganic ion is a type of cofactor. Cofactor just refers to something that needs to bind to the enzyme to make it work. Coenzymes are going to be organic or metallo-organic molecules. So, for instance, heme or NADH, NADPH. These are all FADH2. These are all molecules. Heme being metallo-organic because it's got the iron in the middle. Remember from hemoglobin. And or something like NAD, which is just purely organic, that binds in order to help the reaction proceed forwards. So we covered these terms. So for instance, the metal ions may be required in order to catalyze chemical reactions, so the inorganic ion cofactors or the coenzyme cofactors. Often these are transient carriers, so it's something that holds on to a chemical group for a little while. So for instance, the PLP, which is, here it is, pyridoxal L-phosphate. This is vitamin B6. If you've ever taken a vitamin B pill, you may notice it's very, very yellow. That's the pyridoxal L-phosphate, that's the PLP. And that is really good at holding on to amine groups. And so an amino acid can transfer the amine group to the enzyme, but it's not the enzyme that's holding it per se, but rather the PLP cofactor of the enzyme, which is, it's the enzyme, but it's kind of like the cofactor of the enzyme. It's that part of it that holds on to the amine group, holds on to it, and then transfers it onto a different molecule. So, and we're going to talk about that when we talk about amino acids as one of the last things. But these are all different things, you notice a lot of vitamins. Vitamin B12 is actually cobalt, so it's actually contains cobalt. So it's a metallo-organic molecule. So moving forwards, again, some general terms. The active site is where the substrate binds. The active site it's going to be have a certain environment that's going to make the substrate want to bind. So it may be hydrophobic, which is going to attract the hydrophobic regions of a substrate and help it orientate it. For instance, if this is hydrophobic, well, these parts are going to come together so that this part can then go into the deeper into the substrate tunnel where the chemical reaction may take place. So it helps to bring the substrate in and orientate it correctly. Just to summarize, often chemical reactions of enzymes generally fall into these two types: covalent catalysis and metal cofactor catalysis. The covalent catalysis is where the enzyme and the substrate will be directly bound by a covalent bond as part of the reaction. And so that may be where the substrate binds to the enzyme by a covalent bond, and then while it is bound, that allows other chemical reactions to occur, and then at the end of that chemical reaction it will unbind and then leave as the product. And so an example that we can give is, for instance, hydrolysis of a bond, just the breaking of a bond. Hydrolysis is generally the breaking of a bond that is catalyzed using the breaking of the bonds in water. And so if we have A and B, that's just a carbon-carbon carbon-carbon bond, for example, and we want to break that bond to give carbon plus carbon, for example. A and B want to break that bond. So the chemical reaction that's being catalyzed is the breaking of this bond. And the way that the reaction may proceed, remember that reaction coordinate proceeds forwards, is that you start with A and B bonded together, and you introduce the catalyst X, which really, you know what, that'd be better if it was E for enzyme to make that clear. And so what may happen is that this bond gets broken, and then A reacts with X, and then that gives you A plus X, and because this bond is broken, then it gives you B. But the thing is now that you've done part of the reaction, so you've broken that bond and released B, but A is still bound to the enzyme. And so to release it from the enzyme, typically what happens is that water will be hydrolyzed into H plus and OH minus. And typically what might happen is that the H plus might react with and these are all kind of general examples. This isn't what happens every time, but it's just a worked example. So the H plus might react with the enzyme E, and the OH minus would react with A, and thus that would get this bond would break. And let's say, yeah, so it's going to break, and that then you're going to get release of the A, release of the B now, and the enzyme, you end up what you started out with. So actually looking at it, this is the pair of the pair of electrons is here, so actually this would be attacking here, but you get the idea. The point I'm making is that if I erase everything, the point I'm making is that the reaction is to break this bond here, and the intermediate that is made is that the molecule will bind to the enzyme as an intermediate, and so we call that covalent catalysis, where there's an intermediate where the substrate is bound, either a transition state where the substrate in some form is bound, or part of the substrate is bound to the enzyme before getting released as product. Metal cofactor is different. There can be a range of different things that the metal cofactor can do. Often what it does is that it will stabilize transition states. So there could be if we go kind of back to this slide, So remember what I was saying. So it may be that you have the substrate is the OH in the 2 position, the product is the OH in the 1 position, the transition state is that you have the diol. Now it could be that this diol is unstable and thus a lot of energy is required in order to get there. Now it could be that there isn't an alternative transition state, but rather the enzyme has the same transition state, but because you have a magnesium ion present, the charge from this magnesium ion may stabilize this transition state, and if it's stabilized, well, the energy required to get to it is lower because it's not so undesirable, because it's being stabilized by the magnesium. It is easier to get to that transition state, and then once you get to that transition state, it's all downhill to drop off this OH because it doesn't really want it, even if it is being partially stabilized by the magnesium. And so really easy to drop off and get to that lower energy level of the product. So sometimes the magnesium ion can often help stabilize the transition state, or it may accept or donate electrons in redox reactions for the reaction. So there's a few different ways in which the metal cofactor may proceed in helping the reaction proceed, and we're going to cover that in the glycolysis, TCA cycle, things like that, which is why I'm introducing it now, so that when I talk about it later, it's going to come up. I'm going to test lightly on this in this exam, but when it comes up in glycolysis, I'm going to expect you to understand the mechanism and talk about the mechanism, so I'm kind of introducing the theory behind it now, before the application. There's the enzymes, like I mentioned, I believe in Biochem 1. Enzymes can be broken down into different classes depending on the chemical reaction in which they catalyze. There's 7 different classes because enzymes will do one of 7 different chemical reactions. And that is going to give them what we call an EC number, and that is typically something like X dot X dot X dot X, and the first X is going to be one of these class numbers. And so it's every single enzyme has got a different EC number, enzyme commission, a different enzyme commission number. Every single enzyme in the known universe has got a different EC number, and its EC number is given by the class it belongs to, first of all, then the subclass, then the subclass, then the enzyme number within that subclass. So that's how we all name them all. Sometimes it's referred to, they're parts of these KEGG pathways as well, which you may become familiar with at some point in biomedical research, which are showing metabolic pathways of substrate to substrate to substrate to substrate, catalyzed by enzymes of different EC numbers. But there's the different reactions. I'm not going to go into too much detail of these at this point. But for instance, there's going to be the oxidoreductases, class 1, which are involved in transferring of electrons. Transferases, which transfer groups. We're going to talk about aminotransferases, catalyzed by the aminotransferases, which you have a PLP cofactor, which are transferring amine groups, like it says. Hydrolases, which is transfer of functional groups to water. Lyases, lyase means to cleave, so it's cleaving bonds, typically requiring carbon. Isomerases, which are rearrangements of molecules to get a different isomer. Ligases, which is to stick things back together again. So DNA ligase will stick together 2 pieces of DNA, or a lyase will break it apart. And then translocases, which are involved with moving things around by the different numbers. This is maybe something that may be a bonus point question rather than a big a real question, or not. We've already talked about this, the ground state going up to the transition state. We've already talked about that. Biochemical free energy is something that we're going to talk about more actually in 2 weeks' time. But the biochemical free energy is really the idea that if we start with 1 mole of substrate and 1 mole of product, and we have them both together in a glass vial, and then we add the enzyme, and then we leave it for a while. Where does it end up? Does it end up, if we start with on an equal footing, where does it end up? Like, where does it reach equilibrium? And so it may be that it reaches equilibrium with 0 substrate and 2 molar of product, or it may be 0.2 and 1.8. And you know, you would think that it would just go 100%, but it doesn't. It will go until it reaches equilibrium, and that equilibrium point is called K-EQ. That is the equilibrium point of where the chemical reaction will, if you leave them as one mole of substrate, 1 mole of product, where does it end up? Because remember that these catalytic reactions are reversible; they can go backwards and forwards, and the product can go backwards in some cases to the substrate, depending on the concentration. We're going to talk about that a lot more in the as things go on in the lecture 2 on bioenergetics, and especially when we're going to apply that to things like glycolysis and the TCA cycle, because it becomes really, really important there. But if we so the biochemical free energy change is that if we start with 1 mole of substrate, 1 mole of product, it's going to work to get to KEQ, which is where it wants to sit at its resting point, the ratio of S to P. It wants to sit. And so if it's going to work to get towards there, work is delta G, Gibbs free energy. And so the delta G apostrophe degree sign is the biochemical standard free energy. So it's referring to the under biological conditions, pH 7, how much energy is required in order to get to KEQ. And so we're going to talk about that in a little bit. Activation energy, you're already familiar with. Already familiar with this. Already kind of familiar with this. The rate limiting step is the step that requires the most energy, and so it's going to be typically the step required to get to the transition state. And so by providing the rate limiting step is going to require a lot of energy, so it's going to slow things down. And so if you were to have a different route, then the rate limiting step, the energy required to get there will decrease, and so it will speed up the reaction and push it forwards. So as I promised, this is the thermodynamics. So this is this KEQ and this biochemical delta G, which I'm going to talk about now. I'm going to kind of blast into this. This is going to be mostly new material, and this is going to be becoming really, really important when I talk about applying this to the chemical reactions of glycolysis and TCA cycle and things like that. Okay, so if we have a chemical reaction of substrate to product, like I said, the ratio of the equilibrium constant is given by K, and that describes K is equal to the concentration of product over substrate, whereby, just think about it, just do a little bit of math in your head or on a piece of paper in front of you. If this number is big and this number is small, then this number is going to be big. And so if you have more product and not very much substrate, that's saying that the reaction is going forwards, and K is bigger. If P is big, K is going to be big. On the other hand, if P is small, saying there isn't much product, and S is big, saying there's a lot of substrate, well, that's indicating that the reaction isn't really happening, and so K is going to be small. So if S is big, P is small, K is small. P is big, S is small, K is big. And so a big K means reaction is going forwards. A small number of K means that the reaction is not going forwards and is more stuck at the beginning. Now, notice the little apostrophe here. This means under standard conditions. So that means 1 molar substrate, 1 molar of product. It means pH 7, and it means room temp and room pressure as well, which is this pressure and temp, and they are here. So this is very similar to, this is the biochemical value. This is very similar to when it's actually done in general chemistry. The big difference is that it's going to be at pH 7 to reflect the biological conditions of biology of the cell. And so if we take 1 mole of substrate, 1 mole of product, put them together in a glass tube, probably nothing's going to happen. We then add some enzyme, leave it for a little while, and then see, do you end up with more substrate, do you end up with more product, and that is going to tell you about whether or not we're going to leave it for a while and then see where it rests at equilibrium, and at equilibrium substrate is going to be being made into product, and product is still going to be made into substrate. So it's not at equilibrium. That doesn't mean that it's stopped, but rather that it is now reached the point where the substrate is being turned into the product and the product is being turned into the substrate at the same rate, and so it's steady. It is no longer the total concentration of the two, although it's constantly in flux. It is balanced. It has reached its balance point, where the net gain or loss is no longer changing. And so if we were to leave it for a while, we could then have a look to see, well, how much energy is required in this reaction, which is given by the biochemical delta G. So it's how much energy under biological conditions under these conditions. And so what we can see is that, remember what I said, if KEQ is small, then it's saying that, and let's write it up here, small KEQ equals more substrate, less product. Big KEQ equals more product. And let's I'm going to change color. Let's try a blue. So this is the reverse reaction actually, and this is actually the forward reaction. So big KEQ means forward reaction. You're getting product. Small KEQ, lots of products. So that's the reverse reaction. Now, if we were to look at this table here. Now, if we have a small KEQ, so that's reflecting that actually the reaction went backwards. So you started with 1 mole of substrate, 1 mole of product, and you left it for a while. And if KEQ is small, that's saying that it might actually be 1.8 to 0.2 substrate and product. So that's saying that there's actually a lot more substrate, and the reaction does not want to proceed forwards. Actually, even with the enzyme, the reaction does not want to proceed forwards. The reaction wants to stay mostly at substrate. That may seem weird. That happens a lot, and that may seem weird right now, but we will discuss later, especially in glycolysis, when and why that happens and how it's important for control. And it's to do with fundamentally what this is. It's an equilibrium, and equilibriums are regulated by Le Chatelier's principle. And so that's going to come in later on, but we're going to start from the beginning. So if KEQ is a small number, notice it's 10 to the minus 6, it's favoring substrates, the reverse reaction, and look at biochemical delta G. It is a big positive number, so reflecting that you would need to put in a lot of energy in order to make the reaction go forwards. And generally, how we make the reaction, how we supply this energy is through heat. But you can't do that as a human being. You can't just produce more heat because you would just boil. So it won't work. It's reflecting that actually you're going to have a lot of substrate. And as KEQ increases, right? So this is the small number getting less small, get closer to 1. Notice that biochemical delta G gets smaller until now we're into a positive, or not positive, but a greater, a K greater than 1. So it's getting big. Now we're getting to the point where the number is getting big, and thus product is being made, and the biochemical delta G is now getting negative and more negative. And remember what we say about delta G. When it is negative, the reaction is going to proceed forwards spontaneously. Proceeding forwards spontaneously because you've got a big KEQ. The equilibrium constant is proceeding forwards, making more product. And so what this, which is reflected here. And so what this is telling us is that what we do as biochemists is that we catalog all of the enzymes to find out what their biochemical delta G is. And so we will catalog all of the enzymes. We'll take an enzyme, put it in pH 7 a solution, 1 molar of substrate, 1 molar of product, leave it for a little while, see how much the ratio of product to substrate, and then that's going to be your KEQ. And then from that, you can plug KEQ into here, find the natural log of it, multiply it by negative RT, which is T is room temperature in Kelvin, R is the ideal gas constant. Where's my in joules per Kelvin per mole, and thus multiplied together, they're going to be this. That actually should be there. And so if we multiply this by this, it will give us this. Here, it will give us this. And so what that is telling us, like I said, to finish the thought, is that as biochemists, if we have, we catalog all of these enzymes, get the enzyme, incubate it with 1 mole of substrate and product, see where it ends up, calculate the biochemical delta G, and that tells us under standard conditions, does the reaction favor products or reactants? Now, you may be thinking, well, what is the value of that? Well, that is because that's under standard conditions. How often do you have 1 molar of products, 1 molar of substrate in the human body? It doesn't work like that. It's constantly in flux. And so we're going to talk about this a lot in glycolysis, and so I don't want to go into too much, but I do want to tell you kind of the value of this, is that it may be that the biochemical delta G, so under standard conditions, the delta G is large and negative. And that reflects that no matter what, that enzyme is going to be biting down on that substrate and rapidly turning it into product and doesn't need any help or anything like that. But remember that this is an equilibrium. Substrate to product and the square brackets. Remember, this is an equilibrium. Think about Le Chatelier's principle. Do you remember what Le Chatelier's principle is? Le Chatelier's principle is that if you upset an equilibrium, the equilibrium will work to get back into equilibrium. And so if you have a positive biochemical delta G, reflecting that the reaction is going to be more over here. Now, if you were to, that means that it wants to be more over here. Now it doesn't want to be 100% over here to the left, but it wants to be more over to the left. If you were to, Le Chatelier's principle, if you were to remove product, well, it's no longer, 1000 to 1. It's now going to be 1000 to 0. And that's not right. Le Chatelier's principle is going to say, nope, break down and push the reaction to the right. And so what that becomes important in metabolism, because reaction 1 is changed to reaction 2, is changed to reaction 3, is changed to reaction 4. And so what happens is that reaction 2 will pull the product, because the product of reaction 1 is the substrate of reaction 2. And so reaction 2 will grab that product and convert it into the next product, and in doing so is removing the product from the reaction, thus pushing the reaction forwards by Le Chatelier's principle. Even though it may be that it doesn't actually even want to go forwards, it still wants to go forwards a little bit, and if you remove the product, it's that little bit is going to push it forwards. So it's actually just another level of control for metabolic reactions, which are very, very, very highly regulated. We're going to talk about that more in the glycolysis lecture, but the main thing is that I would like you to understand this reaction here. And what we're saying is that if we take 1 molar of substrate, 1 molar of product, incubate it with the enzyme, leave it for a while, where does it go? And then wherever it turns up, if you feed it into this reaction, it will tell you under standard conditions how much energy is required in order to make the reaction proceed forwards. And that is kind of, and then now we understand what it needs under standard conditions. We can then use that as a starting point to apply it to all of the different conditions that may happen at any one point in time, which is going to be referred to as Q later on, any one point of time, how that may influence where the reaction is going, to understand where the reaction is going at any one point in time. You can then you can say, well, given that I know this, if S equals this, what is P going to equal? We can figure that out, and that's going to come up later on. For now, we're going to we were like the metaphor I like to use. We're climbing up a mountain as things get more and more difficult, and I like to have things where they don't stay up at the top of the mountain for too long, and then bring it back down a little bit to give you relief. We're going back down the mountain a little bit now. We're going to go into some more stuff that's a little bit more elementary and a little bit more to do with Biochem 1. And this slide in particular is, I think, from GenChem, right at the beginning. So you should already be familiar with the idea of the velocity of a reaction is determined by the rate multiplied by the concentration of reactants, substrates. In a first order reaction, this is going to be just one thing, so just S. In a second order reaction, there may be S1 and S2, and it's going to be determined by both. So the speed of the reaction is equal to the rate multiplied by however many substrates are involved. And thus, if you increase the substrate, you increase the concentration of substrate, you increase the velocity. In a way is multiplied by the rate. And the rate is different from the equilibrium. It's a lowercase k, and it's a different constant for that particular reaction. So every reaction has got a rate constant, and the speed, the overall speed, is going to be equal to the amount of substrate that you supply it with. So that you should be familiar with from before. We're going to apply this again to enzymes because it's the same kind of thing. A couple more definitions. I'm actually going to skip over. I'm going to skip over this. You remember that there's the lock and key, and there is the induced fit. I'm not going to cover that here. I'm going to move on from it. But you may want to look over your notes from Biochem 1 and read these slides as well. The idea that lock and key is not the whole picture. It's a part of the picture. The substrate is going to be the enzyme and the substrate are going to be shaped for each other like a lock and a key, but it's not going to be perfect because there's the induced fit. But also, if the lock and the key came together perfectly together, then there's no desire to move forwards. Instead, they come together fairly well, but the intermediate fits really, really well, so it wants to go to the intermediate, but it's a trick because once it goes to the intermediate, it spontaneously breaks down into the products. I remember I used the metaphor in class of this metal bar, and you bend it, and then you break it, and if you get a metal bar and try and bend it like this, it's tough, but if you were to try and bend it over your knee, it's going to be easier, and thus your knee is kind of acting as a catalyst. But if you're try to bend it in like a tube, well, it's not going to be bent because it's got nowhere to go because it's stuck in the tube. But if the tube was shaped like a bend, then the metal bar will change shape to fix that bent tube, and then once it's bent, that's half the battle because then it just easily breaks. That's it in a nutshell. I'm not going to go over it too much. Binding energy, I'm not going to go over too much. This is just really the idea that when substrates bind to enzymes, often it's not going to be, sometimes it may not be a covalent bond that links them, sometimes it may be, but remember that breaking bonds costs energy, forming bonds releases energy. And when a substrate binds, even if it's not by a covalent bond, that initial binding may be by weakened molecular forces, such as hydrogen bonds, and actually forming a hydrogen bond releases energy just in a similar way to a covalent bond. Less energy, but a similar kind of thing. And so the act of the substrate binding to the enzyme does release a small amount of energy that can help the reaction proceed forwards. But as I talked about that before, and it's somewhat of a minor point. Where we're going to get into things, we're going to get into things right now. We're going to get into the meat of things with this, and this is going to be one of the last things I talk about before I conclude this very, very fast lecture. So, remember, substrate, and let's go back to red. Substrate plus enzyme comes together to form an enzyme substrate complex, which then reacts to form the enzyme product complex that then reacts, that then releases to form the enzyme plus the product, and thus you start with enzyme and you finish with enzyme. And so you start and end with the same thing, but what you have done is the net conversion of substrate to product. So it would be very desirable to be able to measure the kinetics at which this occurs, and that's going to be important for a lot of things like drug discovery and understanding the just basic biochemistry of cells and the chemical reactions in cells. Now, if we start off with this graph, you remember this, I did this in class. Start out with this graph. Along the x-axis, we have time. Along the y-axis, we have concentration of whatever. At the beginning, the concentration of, let's say, with the product is going to be low because you haven't made it yet. But you're going to make some product over time. As time increases, so does the concentration of the product because you're making some product. And actually, this here, if we take this, this is a change in concentration over time, and so we have concentration over, let's say concentration of P. That's a really bad P. That looks awful. Let's try that again. Concentration in P of P over time equals, well, what's a change over time? What is a change over time? Think about it in driving your car. Distance over time. speed equals distance over time. So it's something over time. So it's velocity. I'm going to come into that in a little bit, not right now actually, but you get the idea of what I'm talking about here. Similarly, let's use a orange. Similarly, the concentration of substrate is going to start off high, but it's going to decrease over time as it gets used up. So that's so product increases over time, substrate decreases over time, no problem. The other thing we've got to factor in is the enzyme. Now the enzyme, if we've got a concentration of enzyme, we can just call that the total amount of enzyme that is in the reaction. So the reaction hasn't proceeded yet at this point, but how much enzyme do we have? Well, that's going to be the total amount of enzyme. And that total amount of enzyme is going to be made up of enzyme that hasn't bound to anything yet, which is E0, and enzyme that has bound to something. So the concentration of E total is going to be equal to the concentration of the empty enzyme plus the bound enzyme. And so at time point equals 0, at t equals 0, E total is equal to E0, because this is going to be 100, this is going to be 0, which means that E total is going to be 100, so 100 and 100 are the same number, so E total is equal to E0. Later on, when we hit the steady state, E total is going to be equal to ES, because all of the enzyme has bound. And in between, we have the pre-steady state before the enzyme has become completely saturated. In the pre-steady state, it's going to be a mixture. Pre-steady state, it's going to be a mixture of the two. E total is going to be equal to however much E0 there is, the stuff that hasn't had a chance to bind yet, plus the stuff that has had a chance to bind yet. So that's going to be the combination. And so if we were to look at, we'll go back to our graph. We have here this, I'm going to draw in a dotted line, and this is going to be E total. And at the beginning of the reaction, let's go for this dark blue is not going to show up well, is it? Light blue. So at the beginning of the reaction, remember, E total is equal to E0, so it's going to be here. And on the other hand, let's go for a black. At the beginning of the reaction, there is no ES. So ES equals 0. And so it's going to be, concentration is going to be 0. But what's going to happen over time is that the concentration of ES is going to increase as it binds to substrate. E binds to S, and so the concentration is going to increase until ES is equal to E total, reflecting that it's all in that state and it's all bound. And if E is being used up and becoming ES, then the concentration of it is going to decrease. And this point here, let's use our dark blue. At this point here, we can draw a line. And this is the steady state, and this is the pre-steady state. So, and because this isn't changing. It's not changing, so it's steady. It's in the steady state. Now, on the other hand, in the pre-steady state, that's before it hits the pre-steady state, and we have this mixture of ES and E0 making up the total amount of enzyme that's present. Now, what we want to know is actually for this reaction, it can't go any faster, and a product is being made as fast as it possibly can in this brief in this steady state period. But we would actually like to know the rate. Remember the rate of making product. We would like to know what it is in the pre-steady state, because when that happens, you're looking at the rate of this part of the reaction. In these steady state, you're going to be looking at the whole lot, which means you're also looking at how fast the enzyme is able to release the product. And actually, we're less interested in that because typically it's not really a concern in a biological setting. In a biological setting, the enzyme rarely gets saturated, and so sorry, it does get saturated, but in a biological setting, we're not really interested in that so much. We just want to know how this initial part works, because that can tell us things about how to improve drugs and drug binding and inhibitors and things like that. And so we want to measure in the pre-steady state, and you also be able to learn things about these values here. Whereas when you look at the whole of the reaction, it's hard to break it down and learn the individual values. If we just look at it in the pre-steady state, we're able to break down the reaction and figure out these individual values by plugging it into the Michaelis-Menten equation and things like that. So so we want to measure in the pre-steady state. So what we do is that we have some enzyme, we incubate it with a small amount of substrate, and at the initial burst of activity in here, we look at what the velocity is in that initial burst of activity. And that takes, so this is that first graph. And so we can now take that and put it into this second graph, where we look at the product made over time. So that's going to be velocity. And if we look at the velocity at a low concentration, a medium concentration, and a high concentration, well, you'll see that it reaches steady state quite quickly. But if we were to look at the initial points, the initial velocities here, we can calculate, well, what's the initial velocity at the different concentrations, and we can see that notice it is getting steeper as the concentration of substrate increases. And that increase in steepness, more steeper, means more product over time, which means more velocity. So if we look at the initial velocity, we can look at the direct correlative connection between increasing substrate concentration with increased rate. Whereas if you look at it in the steady state, adding more substrate isn't going to make it go any faster. So we're not going to learn about some of these biochemical parameters. And if we were to take the where's my pen? As substrate increases, velocity increases at the V0, meaning initial velocity. If we were to take that and plot that on this graph here as substrate concentration, oh, where's my pen? As substrate concentration increases, so does initial velocity. You can see here, until adding more substrate stops making it go faster. I dislike this graph because really this graph should look more like this, and it kind of gets a little bit I feel gets a little bit confusing by not doing that. But if we were to now you can see that along the bottom, as the substrate concentration increases, so does the initial velocity, which is what we kind of covered before, and until it reaches the saturation point, which is Vmax, which is the fastest that the reaction can go given the amount of enzyme present because the enzyme's saturated. If you added more enzyme, then yeah, Vmax will go up because you've got more capacity for the reaction to go forwards. But adding more substrate isn't going to do anything because it just gets that's limited by the amount of catalyst that you have present. And remember as well that the Km is the substrate concentration required to hit half Vmax. Where if Km is lower, then this is going to be steeper, reflecting that less is required to get to Vmax. Whereas if a Km is higher, then this is going to be less steep, and thus more substrate is required to make the reaction reach Vmax, probably signaling that a lower preference of that substrate for the enzyme. Now, if we're gonna, we're almost at the end. I think there's just let's see, just a few more slides, and I'm not going to cover some of them as well. So, okay. So, if we were to now to define this in terms of algebraically define these parameters, because as we said, this whole reaction of going from enzyme and substrate to enzyme and product proceeding forwards takes part in several steps, and each of these steps is a reversible reaction. There are rates and equilibria associated with each one, and it can go forwards and it can go backwards. And it can go forwards as these positive numbers, and each part has got a positive number, or it can go backwards, and that's reflected by the negative numbers. So E plus S to make ES, it can go forwards as K1, but it can also spontaneously break down as K minus 1. Similarly, ES can go forwards to EP, and the rate of that is given by K2. But also EP could break down and go backwards to ES, and that would be as K minus 2. Now, typically, we can actually largely just ignore this, because product rarely breaks down to substrate again. So we can actually kind of ignore that. And if we want to actually just purely look at ES, and let's use a green this time. If you want to just look at ES, once you reach this point, there's two directions it can go in. Right? It can go backwards, or it can go forwards. So ES can break down in two ways. ES can break down by going to ES can break down either by the K2 reaction forwards, or ES can break down by the K minus 1 reaction going backwards. So ES can break down in one of two different ways. Similarly, E plus S, let's use a light blue, E plus S it can't go backwards, and it can go forwards in one way, K1. So E plus S can go just in one direction, K1. Now, under steady state conditions, the reaction of E plus S coming together, it comes together just as fast. And let's actually use the green for continuity. The breakdown, the formation is just as fast as the breakdown. Well, what does that mean? Well, under steady state, the all of the enzyme is in the ES state, and what it's saying is that as soon as it breaks down in either direction, it doesn't matter. The majority is going to be in this direction. And for the majority of cases, we can, we can kind of ignore this. I don't really want to, but we can kind of ignore this. But as soon as ES breaks down, assumingly to EP, it is immediately restored by the reaction of E plus S. So what this is saying here, this equal sign is saying just as fast as it breaks down, it is replenished in steady state. And that's what steady state is, right? That is where the level of ES stays the same. It doesn't go up or it doesn't go down, because as soon as it does go down in either direction, probably towards product, it stays level, because as soon as it does go down, it immediately goes back up again, because the formation is equal to how fast it breaks down. So that's a really important equation in biochemistry to understand. But like I said, the reaction is generally in the early stages, because of Le Chatelier's principle, we don't have very much P, and so it wants to move forwards to the right, and thus it's generally going to be K1 plus K2. And actually we can kind of ignore K3 as well, because it's going to break down kind of rapidly. But we'll get to that in a moment. So ultimately we're looking at two parts. We're looking at how fast does this come together, and how fast does this reaction making products occur. And those are the two parts of the biochemistry, the enzymatic catalyzed reaction that we are able to glean from the Michaelis-Menten equation, and by measuring these biochemical parameters in these experiments that I was talking about earlier. So if we feed that into the reaction we had before, remember that a few slides ago, remember this that I was talking about? Velocity is equal to K multiplied by the substrate. We feed that into this reaction here. The initial velocity, so the velocity at any one point in time, let's go back to red. The velocity at any one point in time is going to be equal to this part here. So the rate at which this is turned into this, the velocity is equal to however much of this you have, so the concentration of it, multiplied by, where are we? Here we go. The concentration, however much of it we have, multiplied by whatever that K2 rate is. So the velocity at any one time is equal to K2 multiplied by however much ES you have. And however much ES you have is controlled by this part, but whatever it happens to be at any one point of time, the velocity is going to be whatever that is at any one point in time multiplied by the rate. Now we can roll this back a little bit further, right? Because if we ES, measuring what ES is can sometimes be a little bit fiddly. But instead, one time when we do know exactly what, in the pre-steady state, measuring what ES, what number ES is, what concentration it is, can be a little bit fiddly. But the one time that we do know exactly what ES is in the steady state. Now, remember that in the steady state, ES is equal to E total, right? So if I know how much enzyme I put into the reaction, because I'm into my glass vial, and if I know what the Vmax is, well, in that case, V0 when the reaction is saturated with substrate, well, V0 is going to be Vmax. And at that same point, when the reaction is saturated, ES is going to make up the whole of the total of the enzyme. So what we can actually do is that we can take Vmax and E total, where's my pen, and do Vmax, so whatever the maximal velocity is, when it's saturated, if we know how much enzyme is, we can calculate what K2 is. And now, once we know what K2 is, then we can look at, well, what is the velocity at any one time? We know what K2 is, so we can work out what ES is. Or if we know what ES is, we can multiply it by the K2 to predict what the velocity is going to be. And that all feeds into, like I say, the Michaelis-Menten equation, which is relating this all together. The velocity of a reaction is going to be equal to whatever the Vmax is, multiplied by the substrate concentration at that time, divided by whatever the Km is, plus the substrate concentration at that particular time. And remember that all equations have a graph, and so if we were to graph this equation, it's this. It's the same thing. The velocity is equal to the substrate concentration, bearing in mind the Km and the Vmax as other factors into here. So that's how it's all related. Almost done. I think it's just a couple of slides. This I covered last time. So remember that I said that the Km is the substrate concentration when V0 equals Vmax. It is calculated by, and this is the reason why. So Km is really how much S is required to reach half Vmax. And why is that? Well, this is the algebraic reason why, but that's not really super important. I covered that before, and like I said, it's not really super important. Just a final thing to round off. What is Km? Well, like I say, Km is the substrate, is the specificity that an enzyme has for a substrate. If Km is lower, then it's saying that less is required in order to reach half maximal velocity, and we can use, and so that's really telling us, remember that first part of S plus E to make ES, well, the Km is giving us information about how much about this. It's telling it how much it wants to come together, because if the Km is lower, then that's reflecting that this wants to come together more and is moving further to the right. And if you have more of this, then that's going to fill into your velocity reaction of K plus ES. So if Km is higher, you're assumingly going to have more ES, and thus you're going to have a faster velocity. So a lower Km, more ES, more V0. So we can use it to compare substrates within the same enzyme, not different enzymes, but within the same enzyme, to see which is the preferred substrate for the enzyme. And we can see that glucose is a lower Km value than fructose, reflecting that within this one enzyme, hexokinase, which we're going to talk about in glycolysis, less glucose is required to hit the half maximal velocity of hexokinase, and thus we can consider it as the preferred substrate. But remember, ES plus E to ES is not the whole reaction. You also have to factor in ES to EP to E plus P, and thus you cannot compare between different enzymes, because hexokinase ATP may have a really low Km value. However, hexokinase may have a really low K2 value, and so you can't say that one is better than the other, because there are two things to consider. We're going to get into that in a moment. Like I said, this is the Kcat value. Kcat is the K2. They are the same number. So Kcat is K2. They're the same thing. And like I said, this is how we calculate them. What Kcat is reflecting is it's referred to as the turnover number. It is the amount of substrate that is converted into product by one single enzyme per second. So let me say that again. I'm getting a little tired here, guys. The Kcat is the concentration. It's the number of molecules of substrate converted into product per second. So for the enzyme catalase, it converts 40 million hydrogen peroxides to water, actually, is the product. Catalase converts 40 million hydrogen peroxides per second. That's what the Kcat is. Each one enzyme. And thus, if you look at the K total, if you look at what how fast can the reaction work in our how fast can the enzyme turn substrate into product for our whole reaction, which is going to have a bunch of enzyme in it. But if we divide it by how much enzyme we put in it, we'll find it out per enzyme. So if we have our reaction, which has got a concentration of enzyme in it, if we divide it by the concentration of enzyme, we'll find out per enzyme. And so it'll be kind of K2 is actually kind of Vmax, is K2, Kcat is Vmax per enzyme, right? So it's going to be how much is made per enzyme. And the last slide is the last slide. Last slide is when we put these two together. So like I said, Km is the first part of the reaction, Kcat is the second part of the reaction. Smaller Km, faster it goes. Bigger Kcat, faster it goes. And so if we want to, if we consider both of them together, now we can consider enzymes. And so if we say, well, for a particular enzyme, if this is its Kcat, so how fast the second part of the reaction works, so how fast it can turn ES into EP, and then if Km is how fast E plus S can turn into ES, we put these two together as Kcat divided by Km, that will give you this Kcat over Km ratio, and the bigger the number, the faster the reaction is, and you can use that to compare different enzymes. And you can get to the point where the reaction can actually get so fast that it's converting, so it has such a high affinity for a substrate, it's converting so much substrate into product so much that you're actually limited by the speed at which matter can move through, can diffuse through, an interface through liquid. So it's going as fast as physics will allow for some enzymes in particular, like these ones here, which are really, really efficient. So that's so this is really an efficiency, a way of comparing efficiency of different enzymes, which in this case is extremely good ones, but maybe some bad ones as well, or not so good ones rather. So that covers everything I wanted to talk about today. I'm sorry this is an awful lot, but you can go at it at your own pace. I have gone a little fast, but 95% of this material I covered in Biochemistry 1, and there's going to be a quiz on this on Monday. If you need any help, assistance, if there's any clarification, please do reach out. Don't wait till Monday, and we can go through it, and I can explain it at a slower pace with you, and we can conquer it. What we're going to cover on Tuesday is multi-substrate enzymes, and then we're going to go into regulation, and when we talk about regulation, we talk about inhibitors. And we're going to talk about multi-substrate and inhibitors, which is going to be brand new material. And then on the following Thursday, we're going to move into these examples. And we're going to cover a bunch of these examples. And I would like to fit some of them into the exam as well. And the better I cover them, the easier it will be for you to answer. But thank you very much for attending. And I apologize that we're not able to meet in person. But I look forward to seeing you again for this new semester on this coming Tuesday. Have a great day. See you soon.